All right, our class has started. Welcome. And I'm Dave Champlin. And welcome back to a new semester. Welcome to USM if you happen to be new here. I guess the screen doesn't look that bright for some reason. But several of you I don't know, which is cool, because I'm psyched to get to know you. And the first thing to mention is for those of you that are in the lab course, and the lab class is optional. You can sign up for one or both. We're not going to meet this afternoon. We're going to meet Thursday for our first class meeting and look at the bright space announcements for that course. To look at what we're going to do on Thursday a little bit. Okay, so here's what you should do this week. I try to start each Tuesday with that. So you got to get the textbook. We're using the 9th edition of this textbook, and there'll be questions on the exam that aren't covered in our class and are from the textbook. And we'll talk a lot about that because when I'm teaching, I'm always remembering when I was a college student and what that was like. And so I'll say right now that there's a way too much information in our course for you to learn all of it. And so I don't need to have any trick questions or any footnote like super detailed questions on the exam. So the questions that will be from the book won't be a lot. It'll be mostly focused on what happened in class and how the book helps you further understand what happened in class. But there will be topics that are on the exam that weren't covered in our class. And those aren't going to be like the littlest terms and stuff like that. It'll be like a box, a whole page describing some technique. And the question will be, what's that technique used for? Like that, right? Or a figure, and it won't be a little, that little part of the figure, but what's that talking about, like that. And it'll be important for you to bring questions into class from the textbook of things that either we covered in class and you're having a hard time understanding them when you look at the book, or they weren't covered in class and you're wondering what we should know about them. A real famous thing to do is to say, just what's on page 253? What do we need to know? And I'll say nothing, right? That clarifies that. And you don't want to spend a lot of time on a page where we're not going to be expected to learn it, at all for the exam. Let me describe right away this. So this course is called Cellular and Molecular Biology. It's two different courses put together. One of the courses is called Cell Biology, and the other is called Molecular Biology, and they're highly overlapping fields, and actually almost nobody ever refers to them anymore because they've just infiltrated every topic, especially molecular biology has just spread out over all of biology, because molecular biology is the nuts and bolts of life on Earth, and it's universal, so it's just like everything is relevant. And can be described at that molecular level and can be very informative, even if you're looking at ecosystem level events like that. And so it's two different courses, and I'm a molecular biologist. And but I didn't get hired here to teach molecular biology because there was already one and even two professors that taught molecular biology here. And one of them retired, and so then there was one, but still, I wasn't teaching molecular biology, I was teaching other stuff. And then that one left USM, and so suddenly, and left USM pretty suddenly for a job in a private company. And so pretty suddenly, they said, oh, Dave, you can teach molecular biology, and I'll be like, oh man. Yeah, and we'll talk some about how passionate I am about this topic. It's just the best, by far. It's certainly my passion. So I said, sure. So, great, I'll teach that. And it was probably like just before winter break. I had plenty of time to prepare, but I love this so much that I'm like, yeah, I'm all set. I'm super excited, to teach this course. Because I've been teaching a long time, but not this. And so I just went ahead and used the same textbook that the previous professor had been using. And I didn't look at the textbook until like the day before the class. Not because I didn't, need to prepare, or, like that, but I just was like, oh my God, this is just gonna be so cool, like that. And so then I looked at it and I was like, oh my goodness, here are this textbook, and it actually has 8 authors on that textbook. It's not the one we use now, and I'll explain why. Because when I started looking at that book, I was like, this is just exactly what I would say. And I had never seen a textbook like that before, because I never taught molecular biology. And that's different, right? Molecular biologists talk about stuff differently. You can think of a molecular biologist as a car mechanic, right? Well, race car drivers are not car mechanics, and I was teaching classes about race car driving, right? So, those people talk differently. And I thought, oh, I can bring something to these race car driver courses. But in this molecular biology course, the way that book was written, I was like, oh my gosh. And so, and I knew, I had like classes in grad school with a couple of the authors, and 6 of the 8 authors I knew really well. Really well. They'd been my teachers or, kind of almost friends like that. And so I came in first day of class, the first time, and this was like 8 or 9 years ago here, and I said, guess what? I don't have anything to say. So this is how this course is going to work. Now, it's not happening this semester, but it will help you appreciate what's going on. I said, I'm just going to answer your questions from that book, because that book is freaking awesome. I mean, I could talk about it, but we don't use that book anymore for a bunch of reasons. And so only 2 of the students understood, the whole semester, only 2 of the students understood what was going on, and they asked questions, and they guided, the class toward good learning, like that. But I was just like, yeah, that's, great, I'll talk about that, And so even though we don't use the same textbook, and I'm not gonna teach that way at all, it's still true. It's just so true that you should be trying really hard all the time to ask questions. And that's hard to do in class. But when you're outside of class and you look at the textbook, it should become easy to do. Look at the textbook, and when you don't get it, don't understand it, bring that question to class. And then you can ask questions anytime. It's my responsibility to fit them in. They can be, you'll be like, he's not talking about that right now, so I can't, just interrupt and say, well, what were some of your childhood pets? And I'll be like, okay, well, we can talk about my childhood pets for a minute, but then, we'll move on like that, right? So I'll help guide it to fit it in, right? Okay, so get the textbook. That's the first thing to try to do this week, okay? Then we're using the 9th edition. The 7th edition is not good for us. The 8th edition, if you already own it, is fine. The biggest change in the 8th and 9th edition is they renumber and reorganize the chapters. And I often refer to the chapters by number rather than name. And so that would be confusing for you. So it's worth it. The 9th edition has been out for a while. You can find it, you can rent it, stuff like that, okay? Then review the syllabus, okay? So the syllabus is on our Brightspace course under content. And there it is, and I took time already to describe the syllabus in a 9-minute video. You just have to watch it, because there are features of our course that are different than any other courses that you've taken, unless you've taken a course from me. But even if you've taken a course from me, you still need to watch it. And so I'm not going to spend time talking through it. I don't hand it out, because you'll have access to it, the syllabus, all the time on Brightspace. And but it's super important for you to look through it, right? Almost important enough for me to talk about it in class, but also important enough for me to make a video. So you can take time looking through it, and bring those questions to class, right? Like that. So there's just, it's a short syllabus, but it's all serious stuff. And I will talk through this right now. And let's go back to this so you don't have to try to read it. So it turns out that my goal in all the lecture classes I teach is to record the class meeting and then post it on YouTube that day. So we have a requirement, and the requirement is not for you, you're not required to participate, ask a question in class. The requirement is either that you sign the attendance sheet starting next time, so you're here and you're set. If you're here, you're set. But if you miss class, you're required within a week to watch the video, take notes to demonstrate that you participated, and send me pictures of those notes within a week. And there's a whole long list of why that's being done. And the number one is because we have the technology. And the number two is, if you miss class, it's not good enough to get notes from a friend. You need access to the class because we're doing learning like this every time in class. And lots of times we'll only talk about something once and it'll be a big part of the exam, like that, right? We talked about it in class, blah, like that. Three is, I'm supposed to be trying to train you to be scientists. Most of you are not going to go on to be a path to be a scientist, that's fine. But you should be trained as scientists, and here's what scientists should do. They should be skeptical, and they should be thoughtful. And so in class, you should be almost constantly distracted. Going, what the heck? And you're welcome to start Googling. I don't care what devices you use in class. Try not to distract your classmates, but go for it, because I'll tell you stuff and you're like, I never heard that before. I doubt it, or whoa, wouldn't you be able to do this, right? If you could do that, isn't this true, right? So I should give you access to that recording so you can look back to it. Then 4 would be, well, you can go back, even if you attended class, not feeling well, distracted by stuff, so forth, you can go back and watch it that week. 5 is it's ongoing learning. We can't wait and learn and crunch it in during the night before exam. You got to learn this stuff now because we stack it up. This is a very hard course and it stacks up like that, yeah? And 6 is probably some more. But it remains an in-person course. So another requirement is that you try to attend. You're going to miss a couple of classes because of car broke down and sick and stuff like that. But you have to try to attend. And the syllabus paragraphs you have to read because it's a requirement. Attending doesn't give you any points, any credit, extra credit toward passing the course. It's just, we got to do that, okay? And so you got to go through the syllabus because it has a couple of features like that for you to really focus in on and adjust and your thinking, right? And I sympathize for people that are like commuting from Skowhegan, and you're like, how really important is it for me to get to class? And the answer is, well, USM doesn't want me to do the recordings because they want this to be an in-person experience, and it is. And so we have to maintain that. And USM has lots of reasons for wanting students to be on campus, and they're good reasons. They're not mean, making money off of you at the cafeteria, They just want students to be back in an in-person learning experience after, the big COVID pandemic. So it remains an in-person course, but that's a big resource for you to think about and learn about how to use it in this. Then we have a quiz at the end of class on Thursday of this week, and we have another quiz at the end of class on Tuesday, a week from today, and it's based on these videos that I consider to be largely prerequisite videos. And so watch those. The quizzes will be short answer quizzes at the end of class, and that means you'll have time during class to have any clarifying questions. So there are 3 videos to watch for Thursday, like that, and we'll have a quiz at the end of class, yep. And, the first week of class we have a quiz, yeah, because it's stuff we need to get to be all on the same page, okay? Then, there are no makeups if there's a quiz or assignment in class and you miss class. Why not? Because they're low stake. These quizzes and assignments in class are called formative assessments. I'm doing this to see if the students learn, understand that so we can move forward. If not, I should explain, the material more, right? I put it on a video, and I should explain it more. Maybe build an assignment, dive into it a little bit like that, right? And so there are low stakes. We'll have several assignments and quizzes like that, and if you miss 1 or 2, well, you dismissed them, like that, right? Okay, so that's that. And then the next thing is to get the book, read chapter 1, and write down questions, take notes as you go through it. And I acknowledge that that's challenging because we will read the book before we've talked about it, like that. And she'll be like, I don't know any of this stuff. But read through it, take a first pass at learning. That will help you understand more about what we're trying to talk about, set the stage for what we're trying to talk about in class, like that, okay? So if you don't have the textbook quite yet, I managed to put the first 2 chapters we're going to read on electronic reserve on Brightspace. So everybody has access to chapter 1. The second chapter we're going to read is chapter 7. And so electronic reserve is still on that Brightspace under that content where our syllabus is down here, electronic reserve. You click on Aries and it shows you chapters 1 and 7 from our book. And then that's all I can put on, right? Because, right, there's the stuff is copyrighted and I can't just, oh, the textbook's on, you know, I managed to put it online. I can just put a little bit of it online so it can get started. And so here, I'm just going to explain to you, this is the order we're going to go through our textbook, and then we'll loop back to other chapters. And so it's a little bit of an odd looking approach. And what it goes like is this, is there's setting the stage in the first chapter. The first chapter especially kind of sets the stage for the topic cell biology. And we're not going to cover that very much for a while. What we're going to do is go from there to the 7th chapter. And the 7th chapter is really expanding on the videos for the quiz next week. And the videos for next week are all about gene and gene expression, and so transcription regulation like that. And Bryce is back there. Bryce, these are the same videos we watched to start developmental biology, and I'll talk about that. There's a very different focus, so I should just share that with everyone. The videos for next week, I described muscle differentiation. So when we were embryos for the first month of our mom's pregnancy, there were no muscles in our body. But of course, we inherited the genes to make muscles from our parents. We just weren't using them yet. And so that's what those videos are about, is I chose muscle differentiation because everybody has a feeling for what that is. And most of you have learned that your contractile muscle fibers are made up of a set of proteins, actin, myosin, tropomyosin, troponin T, and so forth. Well, those proteins have corresponding human genes that we inherit from our parents, and we just weren't using them, right? There's no heartbeat until about a month after, fertilization. No limb muscles form till about 6 weeks, right? So that's a good choice because you kind of know what a muscle is, and it didn't exist. And it turns out that's developmental biology. I teach that in the fall, that's embryology. And it's a good start there because we're going to talk about how the muscles form. It's a good start here because we're going to focus on the genes, because in those differentiation videos, it's a universal definition of a gene. And that's what molecular biology started and is still primarily about, is genes and genetic information like that. Molecular biology is the union of biochemistry and genetics. And it really got going in 1940s, and then 1950s, Watson and Crick were molecular biologists when they discovered the structure of DNA. So the biochemistry, the physical biochemistry of DNA married to the heritability, the genetics of DNA, like that. And so the gene is a big deal. One of the textbooks I could use for this course is called The Gene, like that, okay? So we're going to go from there, sort of a quick start into the heart of our textbook to section 4. So our book is written in 4 sections. And I think the 4th section is maybe just called cancer. And we're just going to go to these 4 chapters and we'll get those done. And we won't get all the way to there by the first exam. We'll maybe get to there. Maybe 4 chapters, maybe about, on the first exam. We already have target dates. We're going to have 4 exams, 3 exams and a final. And so our first exam is about a month away, like that. We'll probably get through 4 chapters quickly there, okay? All right. So then read chapter 1, take notes, everybody has access to it. And then maybe you consider watching today's video again. You should always do that, right? If you weren't ready for it or there was some part of it. And maybe you don't have to watch the whole thing. Maybe you scroll in because, it was after the fire alarm where you were all distracted or whatever, like that, right? Okay? And write down questions, from that, okay? All right, next thing is that to teach this course topic, I informally call this course Molecular Mechanisms of Cancer. And I'll explain today, why would this be called Molecular Mechanisms of Cancer? Now, some of you will be like, that's great, because you're interested in a career in healthcare. And others of you will say, Not so pleased by that because I'm interested in wildlife biology. Well, we're going to use the molecular mechanism of cancer as just a guide, but the proteins and genes that we'll learn about are universal in life on Earth, like that. Either they're all found in all animals or kind of comparable stories are found in plants and bacteria as well, like that. Yeah. Okay. It's good too because this book that we use now, the author was is a molecular biologist. He retired, who studied cancer his whole life. And we don't have to learn the names of the scientists or the years they did this work. Like that is not the history of science, but we'll use a little bit that help guide us, to help kind of provide some landmark. And so just being aware of the author's career. When he was your age, he was doing some science research in Wisconsin that is still really relevant it's like pivotal, central to our course today. So it all fits together in a nice, way. Okay? Oh, and here he is. Yeah, like that. And so here, this is when he was a couple of years ago. He just retired a couple of years ago. Jeff Cooper. And again, we won't need to know his name, but it's kind of a good way to hang your learning on something like that. You have a friend that likes the Red Sox, and you think of them when you think about trying to remember stuff about the Red Sox, right? It's very effective. So it says our laboratory studies the role of proto-oncogenes. Those are genes that can get mutated and help contribute to cancer formation, like that. And he was a professor at Boston University. He retired and now lives in Gunquit, Maine. And so some of you might know him, might be neighbors of him. It's also really wacky to hear what he does now, because he retired and now he writes fictional medical thrillers, like that. And if you read this, and like a whole bunch of books like that. And so he seems like a really curious character because he's a very, very successful researcher, and now he's retired. And like one of the summary stories is sort of like a student cheats on an exam and the cover-up leads to murder. And you're like, wow, like that, where you're just like, I should probably read that. And I should probably read it to avoid that, because I don't even know who got murdered, but hopefully not the professor. Yeah, so he's great. And here's just some of the other books. There's his book, and then there are several books that are really used in this course. Like I said, one's called The Gene and stuff like that. And we're using this one because it's current. It has a really nice path through it, especially the chapters, the way we take them, okay? So I have a series of things today to cover about what's this course going to be like? And so I described this, and I describe this to every course I teach, but it's something that I just made up, like that. And if you ask other people this question, even you other professors of biology here, you say, if you said biology is a three-legged table, what would those three legs be? Like that, Because it's definitely, and it's like, I made it up, but I'm definitely right. And it's just great. It's just very, very satisfying, like that. And so it's important to think about this. So the table, this three-legged table, is physical, and it's every aspect of life on Earth. Everything is represented in that table. There are no exceptions. It's all in life on Earth. And so these are the actual three legs of biology, like that: physiology, development, and ecology. Okay, and I'll just talk through this briefly because we want to talk about cellular and molecular biology. So physiology is how things are going right now, like your muscles. How are those doing? How's your heart muscle? And also everything else. How's your circulatory system doing? How's your brain doing? Like that. That's physiology. And in this leg, I say physiology and anatomy and something called morphology, the shape of things and how those things function, like shape of bird wings and stuff like that. So physiology is really what is really, filling up your life right now as a living thing, like that. When we're alive, we have physiology, and when we die, that physiology goes away. Also, there's something called cellular physiology. So it's true of all bacteria that they have this physiology. So just the ribosomes and the story of protein synthesis are a big part of cellular physiology. And so that's a huge leg. And so it's important to kind of appreciate the size of these legs. It's just gigantic. because everything that's alive is represented in that leg. And what's it up to from the organismal to the cellular and molecular level? What's it doing? So it's a great big leg. You probably had courses on physiology, anatomy, stuff like that already in college, right? It's a big part of biology. And so, you know, I picked that muscle differentiation for next week's videos because of that. Because, you know, you're moving around. And that's all in that leg. But then, how did it get made? And that turns out to be just a gigantic leg. And that leg is called developmental biology. It used to be called embryology. And about 50 years ago, it started to transform. And it's called developmental biology. And it's just gigantic. And it's hard because it emerged so, you know, everybody was wondering about embryology for all of human history, right? Aristotle, everybody wondered about where did we come from? And there were ideas like that, you know? And what the truth of it is, it's just nobody ever imagined it. And scientists understand it really well now, and it's just exploding. So all of cancer biology fits in that leg. Because cancer is development gone wrong, development out of control, like that. And so development, muscles fit in there too, because when you work out, your muscles get bigger. And that's an issue of developmental biology, like that. And so that's a huge leg. Together, physiology, that leg, and development, they describe everything that ever happens in a living thing on Earth, just those two legs. And you know, again, you're being hopefully skeptical and creative scientific thinkers and think of questions, comments like that. I'd love to talk about them. Yeah. The third one is ecology, and that's the organism in its environment, like that. So there's muscles, right? Muscles at play, like that, in the environment. Everything in the environment, like that, right? So physiology, development, and ecology. If you ask the other biology professors here, there, or anywhere what the three legs are, almost all of them would say, well, one leg is evolution, and that's wrong, and that's one reason for talking about the table, because evolution is the table. It's impossible to point to an example in biology that's not entirely about evolution. And that's because evolution is both a process and a pattern or product. When someone says, oh, you know, I don't believe in evolution, they're talking about the historical process. But if you look at life on Earth, you can see a universal pattern of life on Earth. And the pattern is, at every level, in every detail, it looks like everything's related through a historical long process of evolution from a common ancestor. And that's just a fact, and that is the fact of life on Earth. And you could tell somebody that doesn't believe in evolution, you say that's fine, it's understandable. It occurred over millions of years when we weren't here. But the product or pattern evolution, that's just undeniable. And with the sequencing, the reading of DNA, there's probably more evidence for the history of life on Earth just in DNA than there is for most branches of science, like chemistry. And you'd be like, no way. They've been studying chemistry a long time. It just turns out that DNA is really convenient for computers to gather that information and start comparing it, and it all looks like it's all just closely related. We can easily span the evolutionary distances and times between us and the yeast that causes bread to rise, just easily. Thousands and thousands of times. And supercomputers are doing it all the time. And it's evolution. So it's really important to have a model to describe biology where it's like, yeah, evolution is the thing. And it's physical. The table's physical. Everything about evolution, the product, is physical. Okay? So here's the trick for us. Cellular and molecular biology, those are topics that are off to the side. So we use molecular and cellular biology to understand evolution, but it's not the story of evolution directly. And ecology, development, physiology, like that. So it's tricky to teach. That's the key thing for us. It's tricky to teach this course because I want to talk for a little bit about an example. Well, what's the example? And I'm like, well, the example's in neurobiology, So how many of you taken neurobiology? And the answer is 3, like that. Jeff, he's done, he's ready, Or immunology, right? Or developmental biology, like that, So it's really tough because we don't want this to be a course in immunology, right? You can take that course, but we don't want that. And so it's good to pick cancer because that's not taught in any other courses at USM. So we can pick examples that you haven't learned about because they're examples of cancer biology, like that. And they're super relevant for your health and your family and friends' health and your pet's health. My goodness, right? So, everybody can kind of lead into the focus. And it's about the story of physiology, development, ecology in the context of cancer biology, like that, right? So I wanted to bring that up to show you that it's true for these fields too. It's very hard to teach genetics because you want to use examples and appealing, relevant examples, but to do that, you got to explain them because the students don't know them yet. It's about how antibodies work in immunology, and you're like, didn't take immunology, like that. So we don't have to worry too much about the examples we pick, like that. Even that first quiz, or that quiz next week, I showed the same videos in development, but we focus on a really different angle. So those students aren't, they're not, because I've seen it. I'm sorry, folks that have taken development, they don't get better grades in here, like that, And, I'm sorry, because it's just a different angle, like that, and because we're taking it out there. So there's a bunch of topics like that where you'd be like, where do those fit? And the answer is off to the side. They're explanatory, like that. And so there's a good example, right? This is a video from our quizzes next, a snapshot from the video next week. It's mostly in that development leg, but we're not teaching you a bunch of development, and it's not going to be, you're welcome to ask questions about muscle differentiation. I'll answer them and stuff like that. But that's not what we're going to take on from there. What we're going to take on for them there is that organization of genes and gene expression and what switches them on and off, like that. Here are other topics in biology that are also off to the side. So ornithology is the study of birds. When you're taking a course in ornithology, you're either talking about the physiology, anatomy, morphology of a bird, or its development, or the bird in its environment, like that, and its evolution, right, like that. So, paleontology is off to the side. That's the study of fossils and so forth, like that. So, I just want to run through that for this course, especially for you to appreciate that I'm guiding us through these topics without forcing you to say, oh, these other students had immunology, I didn't have it, I'm frustrated, like that. Yep. So again, molecular mechanisms of cancer, okay? So next thing I want to just describe for a minute is that I'm super enthusiastic about this topic. And that's a book that I ran into in my second year in college. And so when I went to college because my big brother and sister did. I grew up in a little farm town in Minnesota. My mom and dad had gone to college. It was pretty much in the cards, we were going to go to college. And when I went to college, I wanted to study animal behavior. So I wanted to get a major in biology and psychology. So I showed up there, started taking those classes. And the second year of college, I was sitting in the science library, and there were, a place to read, but then there were all the stacks of books, on the shelves. And I saw that book, and I really liked this color. So I went over, and it's a big, thick book. I went over and I picked it up, and I started to read it, just standing there reading it to myself. And after 10 minutes, I said out loud, I'm going to steal this book. And I walked out of there. I could check it out, it's like a student there. But I walked out of it with it held up over the whatever detector system, and I don't know. But it's certainly the only thing I've ever stolen in my life, and and I love that now because I didn't appreciate it at the time, and I was a professor, and even after I started teaching molecular biology, I remember that moment really well, but I still didn't really appreciate that was a passion, right? Everybody says, find your passion, right? And now it's fun because I believe, you found your passion when you're ready to break the law, like that, that's passion, right? But I just so I just shifted, all the stuff I want to do, right? I want to be, this stuff is the best, right? And I love the font of that book. And at that time, nobody was talking about fonts, you know, there's a lot of talking about fonts now, but isn't there just one font back then? And the answer is no, there were like 3. But the one it was in is so good. Oh my God. Yeah, and the author, he was the editor of this famous journal called Cell. Oh my gosh. So I love this stuff so much. That being said, I'm not going to leave anybody behind. It's the opposite. I'm hoping to share that with you, you know, and if you're missing that glowing favored light, you know, I'm feeling bad, right? So it's like that. Next thing is, what this course will be like is, it's the first semester of intro bio. This is what this is. That's how pervasive these topics have become in biology. At every college and university in the United States, the first semester of intro bio is introductory cellular and molecular biology, right? It's got some cell biology, it's got some metabolism, it's got some genetics, it's got some gene expression stuff, right? It's just this, literally, that's it. That's it. So we can think about that, right? Because you know, we're going from there, right? You should have some memory of that. And I just did this. You don't have to see this in detail. This is from the textbook we use here at USM, and these are kind of the chapters we get through in the first semester. And what I did is I wrote some of the topics off to the side, right? And so these are the topics we want to cover. And you covered them all like that. You got, you know, at an introductory level. Problem with intro bio is it just goes so fast that you probably don't remember anything from it, and that's fine. I highlighted these because these are especially chapters that we're like targeting right now, like that. So this molecular biology, that's chapter 7. That's a heart of chapter 7. That gene expression stuff, organization of the gene, like that. So we're dealing with a really big topic, and we're going after it as in a direct, targeted way as we can. But just be aware of that. You know, that you learned all this stuff already, right? It's interesting, it's super interesting to see. Oh my gosh, it's so interesting to see because the instructors vary, right? And like I just said, you know, a lot of instructors, they wouldn't make the same 3-legged table as me at all. Like think of somebody who's a microbiologist teaching intro bio. They're not gonna include any developmental biology, right? There's a topic here, cell signaling, and then the eukaryotic cell cycle. That's just sick in its potential to teach undergrads, because that's what happens under a scab when your skin is healing. And so it's just a sweet, awesome topic to just teach almost all of intro biology about what do you think happens under a scab to make your skin heal, like that. And it turns out we're gonna crank down that path really hard in our course. So that section 4, our 3rd chapter, is about cell signaling. It's about that chapter. If you had intro bio from a microbiologist, you probably didn't cover that chapter because they're like, yeah, you know, even though the evolutionary precursors of cell signaling are there in bacteria, it's called quorum sensing, they're there and they're super cool. It's just super cool. And that's the next thing to say, right? It's not just that I'm passionate about this topic. These topics are super cool, right? It's like that, you know, like that. Like imagine you go over to your friend's house and they have some new music they want to play for you, and they're like, yeah, it's not that I like this, it's super freaking cool. Oh my God. I start to cry in class. Yeah, I'm crying right now just thinking about it. Ha! It's so good. Yeah. All right, so see those topics are into bio, and we're gonna hit them very soon, and it's gonna be a big part of our first exam, and it's hard, okay? So, I don't know why I put this in, just the chapters in the 9th edition. Oh, just to remind you again, if you haven't, because there are a couple of students that already emailed, they said, oh, I happen to have the 8th edition, you know? And I'm like, you can use it, but especially the chapter numbers changed like that. So again, I'll say the cell signaling chapter is chapter 16, you know, and I'll say chapter 16. So again, that'd be a little bit confusing for them. Okay, next thing is starting, well, probably not these next 2 meetings, so we'll have a quiz, but most of the time in class, we do something called a concept chat, and that's in our syllabus. It's described a little bit like that. And so, you know, we'll mix it up, and we won't do a concept chat today, but we'll do one on next Thursday. Then the next thing I have to say is that this is a hard course, like that. I'll just mention that there's this old guy, I can't remember his name. He has a very complicated name, and I think he used to teach some intro physics labs here. and he'd come and visit me. And he came and visit me over break. He just showed up in my lab and sat down and chatted. And he's a nuclear physicist, like quantum physics, quantum field theory and stuff like that. You know, so like one of the smartest people I've ever talked to. And he knows physics really well. And he's interested, he's a broad learner, reader. And he talked to me over the years, and he told me over break, he said, I read that book, that molecular biology book that I recommended to him. He said, I didn't get that. I didn't understand that at all. Yeah, it's a hard course. And I don't see, I'm passionate about it, so it's hard for me to see that. When it's something you're passionate about, you're like, it doesn't seem hard to me, like that. And I'm careful and mindful of that. I've taught this a bunch. But it is super hard. And what I want to emphasize is this is car mechanics, literally. This is the nuts and bolts of life, and this is a course in car mechanics of our body. And from the top down, we're magic. I meditate with these people, and it's just crazy and awesome. From the bottom up, we're just fancy tractors. We're pretty complicated things, but it's just like that. And so all of this is just super physical. It's cause and effect relationships. It's common sense. Like when I say car mechanics, that's intimidating to a lot of people, but all of us understand that if a car doesn't start, no one would get out and check the tires. The tires are needed to go, but not start. And all of this is like that. It's not something you'd be like, well, I guess we'll agree to disagree. It's just like, no. The tires are not needed for the car to start. We don't have to get new tires because the car's not starting, right? Like that. Don't like that. And of course, scientists are open, all excited to disprove anything. A lot of this stuff is, still massive discoveries being made, but a lot of it is just so solid. Like this guy, the author, studied for his career. One of the things he was the first person to discover is this protein called RAS, and RAS is mutated in about a third of all human cancers. Can we disagree about the significance of RAS in cancer? No, like that. Is it ever gonna, be displaced and eliminated as really an important story in cancer biology? No, like that, right? So it's hard. But it is, I like to do this, right? It's a lot of knowledge, but it's knowledge you can hold on to, like that. Just let me mention, again, I grew up in this little farming town. The first time I ever tried to learn anything was in the first day of college. I learned a ton of stuff. I had all kinds of hobbies as a kid, but I did not try to learn. I certainly had no homework all through school, because the school system there was focused on farming. And it was really sweet and very clever people who opposed formal education. Get out and work on a farm, because it's true, you can learn a lot on a farm, like that. So I'd really scramble, and a lot of you are scrambling, and now there are lots of resources to help your learning, to help improve your learning. So one thing you could think about is, it's a hard class, but there are resources, the Learning Center, tutors, stuff like that, to get going with, to help your learning, like that, and help you grow as a learner. It's a good course. Because it's like that, car mechanics. It's like, that's a lot of parts on a bike, right? Bikes are pretty complicated. They're not even cars. And look at that, right? And then look at that. You zoom in on the parts of a bike, and then you're like, this is molecular biology right here, where I'm like, oh my God, I freaking love that so much. Because look at how they are going to hook together, and then they're going to connect your feet with the ground. Oh, shoot, yeah. So here's some stuff. I guess I think I have this a couple of different times. So some good ways to learn for this course. It's physical, so take notes and make little drawings and label drawings, because it's a physical thing. Here's the next thing. I said make little drawings. You should make them big, because that spreads out your learning and your recall. Learning is all about recall. Like, how do you learn a song? You recall it, right? You listen to the song, and then you turn off the music and you sing it. I sang the first part, now I'm going to listen to it again. And the more you recall, the stronger the learning is, the stronger the memory is, like that. And so you got to figure out ways to do that, right? not learning right now because you're just listening, right? You're not recalling it. So it's good to leave class, think about what you just, were told, stuff like that. You get stopped, you get directions in your car. What do? As soon as you pull away, you're repeating the directions as fast as you can to try to hold on to them. And you hold on to them by recalling them and repeatedly like that. That makes stronger memory. So, people focus on athletics and, you got to work out if you want to be good at soccer. But, same is true for learning. And in our society, we just hardly ever talk about that notion. This is a good course for that. So take notes, use the class recordings, make simple label drawings. Every topic is physical. You should be learning and thinking about taking notes. Taking notes, like in class, should not be just like, I'm just grinding this out. I'm just making just documenting this. But think about what you write down. And you're also creating a study guide. So students will also always ask you, is there a study guide? And that is, every class meeting is a study guide, because it's as close as I can kind of distill things and more. But you're making a study guide to help you study before the class. This is a good course for people who like flashcards. The exams are all going to be 50 short answer questions. And your ability to say the words well, explain things clearly, will be evidence that you understand them. And a good way to do that is to make a flashcard. A good question to ask in class is, how would this be covered on an exam? And I would say the question, and then I would say the answer. One thing is, there's always a short, direct answer to everything we're talking about, but we always talk about it in more words. So what you're learning is a more wordy explanation, and it's good to think of, you're welcome to use that on an exam like that, but it's also good to try to pin down what is that exactly, right? Like that, So our quiz on Thursday was of Turns ortholog. What's the definition? And it's a good flashcard tool to just memorize it, because it's a short definition and each word is really carefully used. And if you said it differently, well, that's wrong, like that, okay? So that kind of flashcard mentality is not multiple choice, it's you got to recall it. So, having some flashcards. Here's the next thing that I've never heard anybody else talk about. So I make these things where I'll make a drawing that's big enough to have the labels be flashcards. So it could just be a cell, but make it big enough so the one flashcard is the nucleus, one's the ribosome, one's the cell membrane, or multiple, things like that. So you could both lay them out, and that helps your learning, your recall, because you're like, oh. Like, here's a famous thing about biology, right, is you're talking about the liver, and somebody asks you something about photosynthesis, and you're like, oh my God, no. Not in the liver. That's in plants. That's like outside, right? That's a nice thing about biology, is it's physical like that. And you can be like, we're focused on the liver, and you want to bring up all the liver knowledge, which is over here, like that. There was a student in his class, I think it was last year, who was really struggling. We'll study the cell cycle. It's the 4th chapter we're going to. And, it's G1S, G2M, like that. And it's a circular thing, and there's S, and there's M. And S is when the DNA gets synthesized, and in mitosis, M is when the DNA chromosomes divide, like that. And at a molecular level, there's lots of stuff happening, and it's different. And the student was struggling between paying attention to what happened in S and what happened in M. So, for example, we'll learn just before you enter M, there's a checkpoint that's a DNA damage checkpoint. And there are certain proteins that are regulating that DNA damage checkpoint. And at S, and the entry to S, there's a very different DNA damage checkpoint with different control. And the student was confusing them. And I was like, do you live in an apartment or a house or something like that? And you have a living room? Because just sit down on the couch and say, the couch is mitosis. And that's where I think about mitotic things. And over on the other side of the room, that's S phase. That chair is S phase. I'll go over there when I'm learning about S phase. It's physical, and you want to spread it out. And molecular stuff, you want to bring it up to the real world scale of things. One of the professors who retired that used to teach us was a guy named Tom Knight. Long time ago, retired, I never saw him teach class. But he was a very popular professor, and what he would do is he would have the students get up in this course and say, okay, Chase is going to be the ribosome, And then, you know, Spencer's going to be the mRNA. Here comes mRNA. And then you students are going to be different amino acids like that. Going to come in, and he would have the students physically act it out. And they'd start nodding like, oh, okay, like that. I did that once, and it just got really chaotic because it was the analogy ended up being like a crime scene, and one of the students had a fake gun, and that just went bad fast. So I don't do that, but you can with your friends like that. Make it physical, make it big, and write down questions and confusion like that. So the exam format, again, 50 short answer questions, kind of 2 points each. Sometimes they get bundled together, you know, it's 4 points because it's a, you know, a bunch of stuff we learned like that, and that kind of flashcard learning. Then, you know, we always take linear notes, like right now, we're just, whatever I'm talking about, we're just going through it. But it's good to reorganize those into topics and get them organized and laid out like that. Like, you know, if you're in a chemistry class and you're like, I got this wrong on the exam, and you're talking to the instructor, and you're like, yeah, I thought this was alkynes, and a question about alkynes. And the instructor will be like, oh my God, no, it's about alkenes, you know, geez, you know, and you're like, oh my God, how different are they? One letter, you know, and they're like, oh, you know, and they physically move to say alkynes this is alkynes, and these are alkenes. It's biology, and biology's the best because it's all laid out like that. This is the liver. Those are the photosynthetic leaves over there. So what kind of learning are we going to do? This is a picture from chapter 16, that cell signaling picture. I'll just show you this. We're going to learn a lot of figures like this throughout our course, and we'll learn about this. And you see it's physical. That's the outside of the cell, the extracellular environment. There's a cell membrane. That's a receptor in the cell membrane. This is the cytoplasm. There's the nucleus, like that. Yeah? And people in developmental biology learn some of these, but we're going to learn a lot, and we're going to learn them in a lot of detail. Like, we'll learn all of those, like that. Like AKT. Well, let me just point out this FOXO. The focus of this figure is this protein called FOXO, and we have FOXO in us, and compared to something like hemoglobin, FOXO is way more significant in our lives and the life of every animal. Not all animals have hemoglobin. All animals appear to have this FOXO function. The next thing to say about it is we'll talk about cancer biology, but most of the time we'll really focus on the molecular mechanisms, like the nuts and bolts, not the car ride, but the nuts and bolts that hold the wheels on a car. And that's weird for a student. It's not weird for me, because I just love that stuff. I don't want to ride in a car. I don't care about going to the Smoky Mountains in a car. I just want to look at the nuts and bolts and all the details of the car. FOXO regulates the rate of aging, like that. On the left, in the absence of that growth signal, FOXO goes into the nucleus, and it's a transcription factor, and it's going to activate hundreds of target genes that are all going to slow down the rate of aging. And there are lots of famous animal species that increase or decrease their rate of aging a lot. We don't think of it, but we do a little bit like that. And you know, there's a lot of people now that are interested in that. Oh, I want to slow down my rate of aging, you know. And there are a lot of people taking supplements that are understood to play a role in aging like that, right? And so FOXO is like that, but we won't dwell on that. We'll look at this because it's messed up. FOXO gets into the nucleus to do its aging slowdown job. That's that signal. In the presence of the signal, it gets trapped in the cytoplasm, and it doesn't get to do its job because its job is in a physically different location. Right? Yeah. So just looking at that, we will learn a lot of it. And you could make a big drawing, and you might make a flashcard for FOXO. You might make a little stack of flashcards for FOXO, like that, you know? Because, you know, it will be a bunch of that stuff, okay? So again, best ways to learn, and just a little bit about notes and in the exams. Then next thing is this. So people have studied how students learn STEM topics, science topic. Education research has demonstrated the best way to learn science is by writing and drawing. Lots of students want to take notes on their computers now, you know, like that. So that's been studied. It's just not as good as the old human, old school way of actually getting out and getting it done, mapping it out physically, connecting it with our more familiar real world setting. Yep, like that. And then again, you're making a study guide for these exams, okay? So I just put a slide in that, you know, you should always be taking notes and writing it down. Just, I mean, when you pause, it's not so great to just be thinking. Try to write it, you know, I'm not an artist, you don't have to be an artist, you know. I'm left-handed, you'll see as I start writing stuff in this course, very sloppy writing. But, you know, your notes are for you, really, right? And so, next Tuesday, we'll have quizzes about that topic, like that, lots of information, like that, okay? Again, this shows this. Here's all the detail I put in. I use clay to represent the DNA, the genes in that video, very intentionally, and our big focus is those genes, because it's a universal description of gene regulation. That FOXO, its function is to regulate gene expression. That's its job. Like that. You know, we have hemoglobin transporting oxygen, you know, FOXO transcription regulation, switching genes on all of that's its function. There are about 2,000 transcription factors in our bodies, the human body. And here's FOXO again, just to show you those two parts. It's two parts, you know, in the absence of that signaling factor and the presence of the signaling factor, like that. Yep, okay, so some of that, looking ahead. Then here's a big part of our course. So we have like, well, we'll review some as we go, but they're kind of like 3 big pictures in our course that we just really hammer home. And one of them is that the story of transcription regulation, the dynamic story, the idea that fertilization takes place and the human embryo starts to develop, and it has the genes for the muscles, but it's not making any muscles. It's not expressing those genes. That story is a story that's dynamic. It wasn't making the proteins for muscles, and now it is. So there's a switch, and that's a big part of our course. And with regard to gene expression, the first step in gene expression is called transcription, and FOXO is a transcription factor that starts that. And so a big part of our course is about these transcription factors that dynamically regulate transcription, that switch it on and off, like a light switch. So they bind to the DNA, we'll learn all about it, and then we're gonna have a list. As we go through, we'll come back to this and we'll add different transcription factors to that list. It'll be a growing list, and they're good examples because many transcription factors fit that example, right? So here's the example of FOXO. So there's a different drawing of FOXO, but FOXO is a transcription factor. When it gets switched on, it's going to activate hundreds of genes that do things like regulate cell death, DNA repair, metabolism. They recognize proteins get made that recognize reactive oxidation species and free radicals type things, cell cycle regulation, stuff like that. Here's a figure from a different book that shows, you know, a little worm, a mouse, a fruit fly. They all have FOXO. They call them different, but it's the same protein. It's very, very closely related protein in its amino acid sequence and its function. Here's a person who was the kind of the pioneer of studying FOXO and regulation of lifespan. Her name is Cynthia Kenyon. I just put that up because it's really interesting to see her career. She was at the University of California, San Francisco as a scientist. She was actually studying this little worm, just the most useless worm. It's called C. elegans. And recently, maybe 10 years ago now, she got hired by a company called Calico. And Calico is one of the places where Google is hiding all its billions of dollars. And Calico stands for the California Life Company, because Google is just massively invested, secretly invested in studying the regulation of lifespan and increasing the length of lifespan. If you think about it, our course topic, cancer, that's most often a disease of like older age. There can be childhood cancers, but most typically it's a part of it. And so cancer is really a disease of old age. And this Calico company is interested in just reducing lots of different human health considerations that are correlated with our aging, like that. Yep. So, you know, super fascinating, Foxo, you know, and that Cynthia Kenyon and her life story. But, you know, this is not the course where we go there like that. We're just like, Foxo's cool, because it fits into this. So from this video, we're going to add our first proteins today to our list. And so you haven't watched this yet, but you'll watch it, you know, before a week from now. And the first one is, the first example is a protein called myogenin, and it's a transcription factor that is essential for making muscles. If there's a mutation in myogenin, so it's destroyed, no muscles. And it gets regulated by just getting made. As soon as myogenin gets made, it does its job. That's not true of Foxo. Foxo can be regulated after it's gotten made. So the simplest story is just the transcription factor gets made, it does its job. All transcription factors are proteins, and every organism has to make its own proteins. We make human proteins, and we make human proteins because we inherited human genes. So our Foxo, our myogenin, those are human proteins. They can have very closely related proteins in dogs and so forth, but we make these human proteins. And as soon as myogenin gets made. So myogenin is essential for muscle formation. When does it get made? Just before the muscles form. Does it ever get made in the brain? No. Does it get made in that first month of pregnancy? No. It gets made right before the muscles get formed, yeah? Because myogenin is a transcription factor that binds to the muscle genes and activates them. Says, you've been here the whole time, now it's time to activate, and we're gonna make the RNA and the protein. Okay, so that's our first one. Our next one is Foxo. And so look, there are actually two ways that Foxo is being regulated here. One example is the Foxo can get phosphorylated. So it can have a covalent phosphate added to it. And we'll talk about about phosphorylation lots of times in our course. And phosphorylation just means a covalent phosphate is added by an enzyme, and the enzyme's function is to phosphorylate that target protein. So that's one way that Foxo is regulated. The second way is when Foxo is phosphorylated, it gets bound by a protein that stays in the cytoplasm. So they call that, they say it gets sequestered in the cytoplasm. And so those are really two ways to regulate them. They work together, phosphorylation and the protein binding. But we can actually add two different examples using Foxo. One, again, the first example, myogenin, lots of examples of proteins. As soon as they get made, they do their job. Two, Foxo. It gets excluded from the nucleus. There are several proteins that are transcription factors that can't do their jobs when they get excluded from the nucleus, because their job occurs inside the nucleus. That's our second example, and we can add to that list now. Three, there are many transcription factors that are regulated by phosphorylation. Sometimes they're activated, sometimes they're inhibited. In our case of Foxo, inhibited. When it's phosphorylated, can't do its job, like that. So see, instead of make a list, we'll probably make like 12 of those by our first exam, and that'll be a big part of our first exam, is just transcription factors. When we talk about transcription factors, you know, we're talking about a very specific function. They bind to the DNA regulatory region of genes, and they activate the gene transcription, or they repress it, like that. But they're also proteins, and these are great examples of how proteins can be regulated. There are many metabolic enzymes that can be regulated by phosphorylation, or by sequestering in a physical location, like that. Yep, okay. So here from chapter 2 in our book is describing this phosphorylation activity. So there's a substrate, and this chemical reaction is to phosphorylate that substrate, add a covalent phosphate to that substrate in this very specific location. There's an enzyme that regulates that event, like that. And we're never gonna read chapter 2. Chapter 2 is mostly chemistry, stuff like that. And I'm just like, we gotta rock and roll toward our targets like that. So there'll be plenty of stuff that, you'll have to, pull from some of your previous learning. We have chemistry as a prerequisite for our course like that, genetics like that. And so just to mention that, be aware of that. But you're also, super welcome to ask, any questions about anything at all like that, okay? All right, so I already did that. I already described the first time I taught this course. Again, that wasn't that long ago, and it was freaky, but it's a good thing to have students asking questions like that. Yeah, yeah. And they didn't get what's going on. They did, but it's hard to ask questions. Here, I remember, I don't want to ask questions, right? And it's easy for me to say, oh, ask questions. Did I ask questions in college? I did, but they were just weird, weird questions and stuff. But no, I didn't, it wasn't as good as I, should have been doing like that. One thing about questions is when you're thinking about something, you're totally distracted by that until you resolve it, right? And partly because it's nervous to think about getting ready to ask a question, because what if you, mispronounce a word, or wow, no, like that. Oh my gosh, yeah. And so I talked about that some already. Then I mentioned this. So 2 courses in 1, we're doing great. I mentioned the intro. First chapter kind of jumps into some cell biology, because another, maybe the 4th chapter is actually called the fundamentals of molecular biology, like that. And so we're just going to start diving into some cell biology, and it'll be chapter 1 content like that. So we're going to start doing that. We have another 5-10 minutes like that. And but I should also try to pause. You don't need to wait for me to pause to see if there are questions, but does anyone have any questions or issues about anything? Yep.
But the quizzes, short answer as well?
Yeah, and I should say that, you'll get, you gotta get ready for the quizzes. I probably should mention this after the quizzes. The quizzes aren't as hard as the exam. So we're getting going, we'll have some quizzes, that's good. But I also have felt that, students get a little bit deceived because they're like, oh, that wasn't too bad, But, the quizzes are over focus material that you should already have some familiarity with like that, like that. But yeah, so, they're never, tricky questions, like what's this, what's that, like that. Yeah, but they're like 10 or 12 sort of short answer questions like that. Yep. Other questions people have, everything up?
So is most of our, pretty much all of our material that we're going to be covering in the book?
So yeah, and so the way I describe that is I think that if a student understood everything that happened in one of my classes, they should get at least a B, But to understand it, you gotta look at the book too. And you gotta find the parts of the book that talk about the same thing. It's really important to see that other person, the author, describing the same thing, to hear and see, oh, I see, yeah, like that, right? So there aren't that many points that'll come to the book that are just from the book that I never mention in class. Most of the points will be either points that I talked about really, a lot in class, or are in both the book and the class. Yep. Yeah. So you would want to start with the course material. That's another reason why I record the class, is that's the beginning, and then dive into the book. And another thing is, you wouldn't want to start reading the book like page 1, page 2, page 3. You should look through it the first time, and the section headings, and bold words, and the figures, those topics like that, you want to look at those first, because you're not going to just read it like a novel, okay, page 2, because that's really hard reading, to just grind through 100 pages of really hard science reading. It's good to look at the landmarks and look at stuff and take some notes on those, like that, those section headings, those people try really hard to get those, organize their, writing around section headings. And a good instructor will look at the section headings and say, okay, that's a really important concept. What could I ask on the exam from that section heading? Yep. Yeah, other questions people have? And again, don't wait for me to pause, because I'm not good at that. You just interrupt it. Don't worry about it, okay? So the cell biology is really understanding the different parts of a cell. And cell biology became a topic in about the 1950s with the advent of using electron microscope. So people could see the nucleus and the mitochondria and chloroplast and lysosomes. They hadn't really seen those clearly before. There might have been some experimental evidence for them, but cell biology is really primarily driven by a perspective of eukaryotic cell biology, historically, like that. And it's about describing those parts. And it's also about the idea that molecular biologists can't understand all this stuff, because molecular biology is very reductionistic. It's going to be, we're just going to take it all apart. But if you take the cell membrane apart, you'll lose a lot of your ability to understand the function, of a membrane channel and how things go in and out of the membrane channel. So cell biology is about keeping things together. Then it's really important to also realize cell biology, even in a sophisticated body like ours, it's still the fundamental unit of life is the cell. And each of our cells are really still quite independent. They're collaborative, but they just think, I'm living my one life like that. And so cell biology is great. Students tend to love cell biology, and I think it's because it is exceptionally physical. Like what I mentioned about the channel membrane, there's a protein called aquaporin that lets water in and out across the cell membrane. And there's the scientist that really discovered and studied aquaporin won a Nobel Prize, and there are pictures of him just getting high fives from everybody, because they're like, dang it, this is such a good molecule. And all it does, it's like a donut. It doesn't change shapes, it doesn't regulate water. It can go either way, and all it does is let water in and out of the cell. It's just increasing the rate of osmosis. And I talk a lot about it in intrabiology because it's just such a great molecule. But you're going to really only really understand it when it's in the membrane doing its thing, like that. So that's cell biology. It's like that. And so, you can think about mitochondria, and you learned about mitochondria in intrabiology, a big role in metabolism, like that, like that. So that's some really good cell biology. And I think we got to pause here. We have a couple minutes left, but we'll shut this down. And remember that we have a quiz at the end of class on Tuesday. That content is on Brightspace, and then there's all that other stuff on Brightspace that I mentioned. I will make a bunch of those announcements on Brightspace, and I number them. And every time I make announcements, you get an email notification, oh, there's Brightspace announcement, and then they're there for the whole semester. The stuff I put on Brightspace will be there for the whole semester, so these announcements, like that, I'll try to get this. I should also mention that sometimes the recordings fail, and when they fail, they just fail, for some technical reason like that. But I'll try to upload this to YouTube, and I make a Brightspace announcement. Here's the link, like that. So ever onward. Okay? Yeah, all right, awesome. That's it. Thank you.